We're going to go out to the Magoon Research Center, where we've had a video produced about the research center, and then afterwards we'll have an opportunity to have some questions and answers about that. Dr. Baldos and Dr. Amore, are you still there?
Yes, we're here.
Yes.
Great. So I think we've luckily been introduced to you. Should we go ahead? We'll go ahead and watch the video now and then have some questions afterwards.
The Magoon Research Station is a research and instructional facility for the University of Hawaii. Located in Manoa Valley near the university's main campus, it is operated by the College of Tropical Agriculture and Human Resources. The college, also known as CTAR, has 5 departments and a staff of scientists, instructors, and technicians.
Aloha, my name is Orville Baldos, and I'm an assistant researcher at the Department of Tropical Plant and Soil Sciences here at UH Manoa. So today we are going to talk about Magoon Research Facility in the heart of Manoa Valley. So the Magoon Research Facility was actually a piece of land that was previously owned by the Magoon family, hence it was named after this family. This part of land was purchased by UH in 1965 through land grant funds and legislative funding. In 1967, this was identified as a place that CTAR's research and extension and instruction activities, a facility for these activities would be built. The construction of the facility was completed in 1976, and this facility had an impact on the students because it is a close enough place for students in agriculture to do their field research. So it's just walking distance from the Manoa campus. So the mission and function of the Magoon Research Facility is basically to assist in the teaching and the research activities of faculty and students of UH Manoa. The facility is located on Woodlawn Drive, and as you go to the driveway of the facility, you are then in front of the head house. So the head house contains the manager's office, there's a conference room, there's also a small lab in there, also a restroom facility, storage for heavy equipment, a tool room for garden equipment for class use. We also have a kitchen, a small kitchen, and a dryer for drying samples of plants, and also a cold room for storing seeds. The left side contains a room wherein heavy equipment like golf carts are stored. On the right side is the conference room where students have their classes.
We also do distance learning activities here in the classroom because this is equipped with webcams. In addition to instructional use, we also use this conference room for industry groups so that we can serve them in terms of having their board meetings here, and also for the master gardeners.
So as you exit the head house, so you have tents that are used for research and also for instructional activities. So students can do their potting activities here or repotting. People can also do informal lectures in these tents, so it's more open air. And beyond the tents, as you can see here, are the turf demonstration plots. And over here is the Wi-Fi enabled weather station that collects data on rainfall, temperature, relative humidity, and this could be easily accessed through a website. And then around the head house are also ornamental plantings that are used for instruction. So we use these plantings for the plant propagation class, also in the horticultural practices class, and also for the foliage and flower crop production class. To the right of the turf demonstration plots is the greenhouses or the glass houses. So the first greenhouse is the instruction greenhouse. So this first greenhouse is used by a number of TPSS classes. This greenhouse also contains a succulent collection for instructional use. So this is used in different classes, again, for 420, which is the plant propagation class, the horticulture class, and also the foliage and flower production class. At the back of Greenhouse 1, you have a propagation area equipped with mist settings. There's also a Wi-Fi enabled temperature sensor at the back area that you can also monitor temperature and humidity. The next greenhouse is Greenhouse 2. You have basically research activities going on in there. One of the research programs that are housed in that greenhouse is Dr. Melser's biosecurity research. Dr. Mansart's papaya research is also housed in that greenhouse. And there's also some bench spaces in that greenhouse that you can rent for a fee. And there's another bench there that's equipped with supplemental lighting. So this is used for photoperiod experiments, mainly for instructional purposes. The third greenhouse is also a research greenhouse, and this greenhouse houses Dr. Lincoln's indigenous crops research. And Dr. Lincoln mainly works on sugarcane, banana, breadfruit, and also kava. To the left of the greenhouses is Shadehouse number 5. And Shadehouse number 5 is a mix of instruction and research. So this Shadehouse is also equipped with Wi-Fi enabled temperature sensors, wherein you can access that also through the internet, or if you can also use it, you can also access temperature data through an app in your phone. And this Shadehouse contains the ornamental germplasm collection of Dr. Criley, and also some of my native plant collections. And then the rest of the space is out for rent, so depending on if there's any student or faculty who need space for doing experiments, this Shadehouse is available for that purpose. Then you have Shadehouse number 6, which is the next Shadehouse on this side. This Shadehouse is where Dr. Amore's dendrobium stock plants or mother plants are located. And then Makai of the Shadehouse, so there's another Shadehouse on this side. This is Shadehouse number 4. Shadehouse number 4 is where the Anthurium breeding program is housed. So this is where the stock plants or the mother plants and also the hybrid plants are located. Shadehouse number 7 is this Shadehouse here. So this is both used for instruction and research purposes. Again, there is another temperature sensor in there that's connected to the internet for monitoring. And this Shadehouse also houses ornamental plant germplasm that's used for instructional purposes. Also, native plant germplasm that I use, particularly the peperomias that are used for indoor. About half of the Shadehouse contains the dendrobium breeding plants of Dr. Amore. And every fall semester, the other half of the Shadehouse is used for poinsettia production. So this is mainly for the TPSS 364 or the Horticulture Theory and Practices class, and also the flower and foliage production class. So this is done every fall semester. At the back of the Shadehouse, you have this Hoophouse. So this is mainly used for instruction or research purposes. This is equipped with a mist bench at the back, and the benches in front also contain drip irrigation. And every fall we use this facility for the poinsettia production. So we have probably around like at least 400 or 500 plants, and if there's not enough space in Shadehouse 7, then some of the poinsettias will go into this hoop house. At the back of the hoop house is the outdoor benches, which some of it will be for rent. So if you're interested in doing research, if you're faculty or a student that's you need space for research, then this could be a place that you could do your research on. Some of the benches are also occupied by my research program, so I have the native plant germplasm in here, and also Dr. Lincoln's indigenous crops, so mostly breadfruit. And then the GSO, or the Graduate Student Organization of the TPSS department, also has a bench for their purposes. So if they're doing plant sales, so they have a bench for growing plants. All of these benches at the back are sprinkler irrigated and are rain fed also, so it's not covered. Mauka of the Magoon head house are the glass houses of the Plant Environmental Protection Sciences. So these glass houses are managed by the PEPS department, and this is where they do their disease studies. Beside the PEPS greenhouses is the student beds. So these beds are mainly used for instructional purposes. So this is where students in the TPSS 364, the Horticulture Theory and Practices class, grow their vegetables or flower crops in the fall semester. And this is also where the weed science class also do their experiments on weed control. At the back of the student beds are the fruit tree collection. So we have some mangoes in there. We also have avocado varieties, citrus, and also banana. You also have a separate glass house here, which it could be rented by faculty in the department or outside of the department for a fee. In front of the student beds are collections of ornamental trees like the cut foliage eucalyptus, which we just planted about 3 years ago. And then we also have a plumeria collection. This is Dr. Cridey's plumeria collection planted. Further east of this area are our storage spaces for instruction and also for research use. So this is where we keep our pots and fertilizers. At the back of the storage area is the animal facility. So this facility is managed by the Human Nutrition, Food, and Animal Sciences Department. Makai of the Magoon head house, there are 2 sets of glass houses. These glass houses are managed by the Natural Resources and Environmental Management, or NREM department. So these are where NREM department students and also faculty do some of their research. And then further Makai of that area is the Student Organic Farm Training Farm, or soft farm. So this is a soft farm here. And then there's another area here that's managed by the TPSS department for research and also for teaching. On the right side of the picture here are also field plots that are managed by the NREM department. So that basically is a short field trip, I guess, virtual field trip of the Magoon Research Facility. And now I am going to present to you my research program on native Hawaiian plants. So my research program at UH Manoa, my aims is basically to collect, select, and evaluate non-endangered and underutilized species for a number of ornamental purposes. So I look for native plants that can be used as landscape plants, as potted plants, as an indoor or as a house plant, and also as a cut foliage plant. So why use natives as ornamentals? So in the United States, the use of native plants as ornamentals is gaining popularity. People are more aware about environmental effects or of plants, and we are now more. It's more than just aesthetics nowadays with using plants in the landscape. So some issues that people are more aware of now is water use and conservation. So we want to use native plants in order to conserve water. Native plants also contribute to biodiversity conservation. It also helps mitigate invasive species spread, because if you're using native plants or plants that are native to your region, you're not introducing any plants that come from other regions that may become invasive or weedy. Another issue that can help, native plants can help with, is stormwater management. So some of the benefits with native plants is that it's a habitat for pollinators and other wildlife, and it also provides a sense of place. So here in Hawaii, native plants have been promoted in the last 28 years. So there was a law that was passed in 1991 that is known as Act 73. So this act or law is trying to promote native plants in publicly funded landscaping projects. So this has been in place since 1991. So in 2015, Act 73, which was passed in 1991, was replaced by Act 23315 relating to Hawaiian plants. So this state law gives a little bit more teeth to the previous law by putting in numbers. So it aims to increase the plant footprint of native plants from 10% in 2019 to about 35% by 2030. So despite promoting native Hawaiian plants, it's still not yet, I guess, the use of native plants in Hawaii is not fully realized because of the following bottlenecks. So there's still a need to increase availability of native plants. We need more variety, specifically like more textures or more colors of native plants. There's also a need to improve horticultural information because not a lot of these plants are commonly cultivated. So one of the reasons why people are not planting natives is that they don't know how to take care of them. So we need more horticultural information. Currently, there's little to no development of selections or development of nativars that are specifically used for landscape and indoor purposes. This is how we develop our selections for natives. So the first part of native plant development for ornamental use is germplasm collection. So what we do is we get propagules from wild and cultivated sources. If we collect plants from wild populations, we get permission from the landowner or the state agency. So we get permits first in order to collect these plant materials for wild collected species. So our target species for germplasm collection for landscape use is basically the coastal area. So most urban areas here in Hawaii are located in lowland areas. So we would like to match the plants that we evaluate with where their final, I guess, destination is. And then our target species for indoor use are areas that are shady, mesic to wet, about 300 to 1,250 meters above sea level. So this is a bit more higher elevation species. So here are just a few pictures of native species in the wild. So this is Lipochita integrifolia or nehe, which is a potential ground cover for landscapes. So this is a picture of it in the wild on the coast of South Point on the Big Island. This is Jackmondia sanvichensis or Paohiiaka, growing on a sandy beach on the Big Island. And then these are Peperomia sanvichensis plants that are found on West Oahu, on the higher elevations of the Waianae mountain range. So you can see here that they grow well under boulders, on boulders with moss-covered boulders. And then you also have Peperomia tetraphylla, which is growing on either moss-covered boulders or moss-covered trees under shade. And then you have Peperomia mauiensis. So you can see here the plants growing in the wild, usually on cliff sides, rocky with moss growing on them, and also under the shade. So the environment wherein the plant actually grows gives us an indication of how it's going to be used in, like, for example, the landscape or maybe for indoor use. So during collection, we document the necessary information like GPS coordinates. We take photographs of the plants in situ or in the wild. If we collect the material from botanical gardens, then we record or we get information from botanical garden records. So whatever they list in terms of information, we also get a copy of that. And most of our collections are from stem cuttings. We get stem cuttings and then root it in the greenhouse and then pot it up. In the case of Peperomia, we have moved our propagation indoors because of the heat. Peperomia seems to be rooting fairly well under AC conditions. Well, because they're mainly a higher elevation species. And we also tried growing the stock plants under hydroponics, just to increase the plant material faster. So once we have these collections or plants, we do an initial evaluation for ornamental characteristics. And we also do studies to develop propagation protocols. In order for a native plant to be successful as an ornamental, it has to be easily propagated. So in the propagation studies, we test whether rooting hormones can improve rooting of these cuttings or not. We are also looking at trying to maximize the number of cuttings per stem, because initially these plant materials are very, very limited in number and we want to increase it pretty quickly. So we look at studies to maximizing the number of stem cuttings that we can harvest per stem. We also look at mist versus sub-irrigation conditions, and also put plants in micropropagation or tissue culture, and then look at ways to acclimatize it. We have put Peperomia species in tissue culture and we produce these micro cuttings that are unrooted. So we did a study recently on acclimatization. So using these micro cuttings that don't have any roots, treating it with rooting hormone and testing what sort of growing media would work best in terms of rooting them. And then for plants that have dormancy in them, then we do some studies to hasten or break dormancy of those seeds. Beside the propagation studies, we also look at container evaluation studies. So we grow the accessions up to a certain size and characterize them, looking at color, leaf number, maybe flower number, also hairiness, that sort of thing, and also some ornamental characteristics. Then once we do the characterization, we select what the best performing selection is. In our indoor light tolerance studies, we grow the accessions to size and then put them under different lighting conditions.
So these racks have fluorescent bulbs in it with different lights. So we put the plants in here and observe the plants for 6 months. So look at the growth, also the quality. And we also have relative humidity loggers and temperature loggers here, plus light loggers to make sure that we're getting the right conditions for indoor use.
If the species or the selection performs well in low light, high light, and then office light, then that's a good selection for release as an indoor plant. So recently we have released selections for landscape and also for indoor use. The ones that we recently released for outdoor or landscape use is Jackmontia sanvicensis Puhalabe. So this particular selection came from Maui via the Maui Nui Botanical Gardens. So this selection has blue flowers, has hairy leaves, so the leaves are hairy that it casts a silvery tone to it. So that makes it ornamental. For this selection, we recommend using as a ground cover in landscapes or also as a container plant. The other selection that we released this year is Peperomia sanvicensis palichea. So this selection is a Peperomia species that's ideal for indoor use, mostly for small containers. So they work well in like 3-inch pots, and they can withstand or tolerate low light conditions even at the lowest light levels. So that's my research program in a nutshell. And this last slide, I just wanted to share with you basically a personal note on how I became a horticulturist working on native plants. So growing up, I had parents who had advanced degrees in agriculture. So they're working in research fields. So this basically exposed me to horticulture at a very young age. Having a small like garden plot allowed me to grow plants. And at high school, I had an interest in collecting more ornamental plants. So I love reading on my spare time. I love reading like photo books like Tropica or Hortica or Exotica, and basically identifying the plants by scientific name. And that was, I guess for me, it was fun. So that plus the collecting was made it enjoyable. So this sort of hobby of collecting plants, I guess, directed me to pursue agriculture as a degree and specializing in landscape horticulture. And after college, I was introduced to natives, particularly native Philippine orchids, as a research assistant. And this inspired me to pursue advanced degrees in the field, particularly looking at natives using natives for landscapes or for other ornamental purposes. So this is me, the young me, looking for an orchid in a mangrove forest in the Philippines. So the orchid that I was looking for is this orchid here. It's a Euphlebium papilionaceum. So it was a new species at that time. So I was part of the research team who was looking for this species and trying to describe this as a new species to science. We were able to find the orchid and collected samples for further research. And if you would like to contact me, these are my contact information. So you can email me at obaldos@hawaii.edu. You can also check out my lab website. So this is the URL for my lab website. And you can also follow me on my Instagram page. So my Instagram handle is UH_ornamental_hort_ and then lab. Thank you.
Hi, I'm Teresita Tessie Amore, an assistant researcher with the Tropical Plant and Soil Sciences Department of CTAR. I do anthurium and flower breeding, and I've been doing this kind of work for many years now. Developing a new anthurium variety requires quite a bit of patience, as a new variety is named and released about 14 years from pollination. The two main phases are hybridization, which takes about 2.5 years, and the lengthier selection and evaluation phase, which, including the field test phase, which is roughly the time of the Anthurium crop cycle, will take about 10 to 12 years. We normally associate the Anthurium flower with two botanical parts. The spathe, which is the more colorful part, is a modified leaf. The spadix is the botanical flower, and it is a complete flower. However, it is protogynous, meaning the female parts mature before the male parts.
The first step is to hybridize or make a new cross between two desired parents.
The hybridization phase takes about 2.5 years from pollination to selecting a seedling. Although anthuriums have natural pollinators, such as the gecko or ants, as a breeder, we select the parents for breeding with a goal in mind, whether it's color or flower shape. We take pollen from the male parent and with our fingers, transfer the pollen to the receptive stigma, and in a few weeks, wart-like structures indicate fruits are starting to form.
It takes about 6 months to generate a fruit or to produce a fruit from which we have seeds to germinate. These are anthurium seeds on the plant. As you can see, it's a berry and it may contain 1 to 2 seeds.
Here are close-ups of the mature pollen, receptive stigma, developing fruits, and the mature fruits. When the berries are mature, we extract the seeds from the fruit in a container. Out of curiosity, I estimated the number of seeds from a single pollination. Anthurium berries produce 1 to 2 seeds per fruit. In this example, 173 fruits were formed, and we were able to get 256 seeds.
After we squeeze out the seeds, we lay out the seeds in a pot filled with a suitable germination medium. The medium can be anywhere like orchid bark or any of the potting mixes that are commercially available. So this is about 2 weeks old, and you can see that the seedlings are starting to germinate.
When the seedlings reach a suitable height, they are moved into flats and then pots to observe flowering. So here we go. These are the seedling selection stages.
We grow the seedlings in 1-gallon pots and watch them flower and observe for traits that are important for industry. For instance, we find these seedlings and we look for yield. Ideally, we look for 6 flowers per plant per year. We will come over and look at these plants and try and take weekly data.
As the seedlings start to flower, we make individual seedling selections based on what we thought were traits important to the grower. Seedling selection on spathe color was based on what the breeder thought was unusual or fulfilled what the commercial growers wanted to produce and market. So on the bottom left, this is something that's undesirable because of the furled spathe. On the upper right is something we thought was desirable. Over the past few years, we have been soliciting input from the designers during the selection process. The designers have helped us identify selections of interest to the industry that have unique traits, such as spathe color and spathe conformation. So we went over the hybridization process that takes about 2.5 years. We will now discuss the second phase, which is the selection and evaluation phase. Once we select a particular seedling, we then take an immature leaf and clone via tissue culture and multiply for testing for advanced testing or field testing with our growers cooperators. So for the tissue culture phase, we choose leaf tissues to speed up our process. We hope to get plant materials to the field within 2 to 3 years from initial selection.
So here's the starting leaf material on the upper left, and we plate it onto petri dishes, observe over time until shoot-like structures appear, such as what you see on the bottom left. Then we move them into larger containers to produce stock plants for multiplication in tissue culture. The rate of shoot development can vary, as you can see on the left, which has a few shoots, whereas on the right there's a lot of shoots, or mostly cell mass, that will eventually form into shoots. Shoots are then cut and placed on a suitable medium. We produce anywhere from 200 to 1,000 plantlets to test with cooperators, depending on the selection's potential for commercial cropping. Large numbers of plant materials for field testing are needed so that each cooperator or site has adequate numbers of plants to observe the selection's performance over time. We also index for bacterial disease caused by Xanthomonas axonopodis, Pathovar dieffenbachii. In our tissue culture collection, we maintain selections that are sent out for cooperators for field testing, such as the 2 boxes on the left, which are to be evaluated by our cooperators. The middle box is a selection that is going to be released very shortly, and so plant materials such as these are sent to cooperators so they can contract with laboratories so that they can produce the plant materials for commercial cropping. The box on the right is an example of our germplasm that we keep. That is, these are the released varieties that we keep in case growers want to have them ready for commercial cropping. Advanced testing takes place over several locations or cooperator sites. The individual seedling that was cloned will now be evaluated for production qualities, such as what's listed here. Growers also do test shipments to see how well the flowers handle the post-production life. Does it ship easily? Is there bruising or damage? Is the product acceptable? Or can they be used in designs?
This is UH2237, a seedling selection between tropical and tropic fire. This is a selection that we have been conducting advanced testing with our cooperator growers on the Big Island. Our cooperator growers provide us with production data, packing data, and my colleague on the Big Island, Joanne Lichty Imamura, does the vase life studies, where she looks at how long these selections last with or without BA. After 5 to 7 years with advanced testing, then we are ready to release a variety. So this variety is, we have gathered enough data that we can release it by the end of the year.
When we think about the Anthurium varieties that have become widely successful and widely accepted by the industry, the pink Marian Seaforth was before Magoon was built, because Marian Seaforth came out in 1963. The other varieties that are notable or that have come out since have been developed at Magoon, and this includes Lavender Lady, Regina, these are all developed at Magoon, Tropic Fire, and I would say that right now the #1 white that's being grown is Hokuloa, UH1349, and these were all developed at the Magoon greenhouse facility. In 2007, there was a renovation of the Magoon greenhouses. So the orchid section moved out of those glass houses, which were considerably hot and dry for the orchids. So we moved to Saran Shadehouse, and we also had a section that was open on the sides with a polyphylon roof. And that was better for the orchids because it was cooler and we had
more air circulation. When we evaluate dendrobiums for cut flowers, we take the flower quality, such as spray length, number of flowers per spray, and we also look at the vase life. In the early 1970s, that's when the seed propagated dendrobium varieties were developed and were released. So at the time, the first one that came out was UH44 Uni-Y Blush. Then came UH232, which is widely grown now, still widely grown, UH232 Uni-Y Supreme, and UH306 Uni-Y Pearl. However, Uni-Y Mist, which is UH800, that was developed in the late 90s at Magoon. So when we think about the number of varieties that have been tested, a number of the successful ones will trace their origins or roots at Magoon. And so that includes UH1081 Uni-Y Royale and UH1427 Wally Murazani. As of recently, I would say that the top white is UH800 Uni-Y Mist, the top pink is Uni-Y Supreme UH232, and the top purple is UH1427 Wally Murazani. All have had their roots, all have been developed at the Magoon Greenhouse facility. So indeed, when we look back and see what varieties have been successful, the Magoon Greenhouse facility has been very valuable in developing all these varieties that are used in the floral trade. Over the past few years, our graduate students and undergraduates have been working at the Magoon Greenhouse facility to produce poinsettias during the holiday season. So in the fall, the students receive cuttings, pot them up, groom them, and grow them out so that they can be sold to the university community in early December. So the students learn how to grow, market, and sell a seasonal product. Every summer until COVID, we had the Ag Discovery program. The theoretical part is in the conference room, and then we take them out to the greenhouse. In August of 1985, the graduate program, which was then called the Horticulture Graduate Program, celebrated its 25th year. The Magoon Greenhouse facility was used as the venue for the alumni luau, and we had a tent, an imu, and a stage where a hula halau performed.
The Magoon Research Station in Manoa Valley continues to provide an ideal setting for research and instruction. The station helps fulfill the mission of the College of Tropical Agriculture and Human Resources to create and deliver knowledge that supports and strengthens families, agricultural and food systems, and the natural environment.
Well, that was an incredible tour. Thank you guys so much for that amazing video that you did. That took a lot of work, I'm sure. So I have a quick question. Are you guys open to the public? Maybe Dr. Baldos, you could take that one?
So we're not really open to the public, but sometimes we have, I guess, open tours, maybe. The legislators also come in and, I guess, schedule a visit. But now that it's COVID, it's, yeah, we're kind of like closed right now.
Yeah, as a lot of things are, and we're used to that. But and what came up a lot in the growers and breeders forum that we kept hearing, especially from Dr. Keach and from Russell and whatnot, was one of the biggest issues, obviously, with everything, but was the funding. And I was wondering where you guys get your funding from.
So we received funding from number of local sources. Primarily, HFNA has been instrumental in helping us by asking the state legislature to provide funding to our grants. Secondly, we had a grant with Hawaii Department of Ag to develop new germplasm. The grant does not cover only ornamentals, but includes other commodities such as sweet potato and taro. But there is a portion of the grant, I'd say about half of the grant is for flowers and primarily flowers and native plants. So Orville's on the grant also. So that's how we get funded.
Yeah, and some like small grants, like under $10,000, I guess. It's just for buying supplies for research.
Anything on the native side? Do you guys get funding to support the native?
It's only the germplasm grant that the HDOA had. So I haven't explored other options like DOT. Maybe they might be interested, the Hawaii Department of Transportation. I've tried applying for grants in the mainland, but it's not, well, since their focus is a little bit different, it's a little bit harder to get funding from the mainland granting agencies.
Yeah, funding is always going to be our challenges. Well, maybe Oprah or one of the billionaires who keep buying up all the islands can help to support the native plant funding as well for you guys. I'll put a good word in for you. Somebody asked, do you guys take internships at all? Dr. Amore?
We would love to. We all have, it depends where and when. In the past, we had interns. In fact, Dr. Keach was an intern with us when he was doing his undergrad. Where's Dr. Keach? He can verify that he did work for us well. He was here in Hawaii for about a year while he was going for his undergraduate. So we do have internships. Or we can work and have internships for interested students to come and work with us.
We can also do, I guess, internal ones for our own students.
Yeah, it sounds like something that a lot of younger folk would be really interested in. I know that a lot of people come and they want to grow native plants. I grow Dracaena, but a lot of people I know, it's all native foliage for landscaping plants. It's very popular on the Big Island. So maybe that'd be a great way for somebody to come through your program and then have a great opportunity after that to create a business. So how close to Magoon is the UH Manoa campus?
So it's basically a 10-minute walk from campus. Yeah. If you drive it, I guess it's a little bit longer if you drive, because it's the road is a little bit longer.
It looked really exceptionally clean and really well maintained. Is that because you have a lot of student help, or are you guys just exceptionally clean greenhouse folk?
I'd like to answer that. We have excellent ag techs who help us with that. And we also do have some students who work with us to maintain the plants in our greenhouses. But mainly what you saw in the video was the handiwork of our ag techs. And at the time the video was shot, I believe, and Orville can corroborate, we had 2 full-time ag techs, and they're the ones retiring at the end of the year.
Yeah.
Oh. That's the great migration we're having right now. What classes are conducted at the facility, Dr. Baldos?
So I teach the 364, so you've seen in the video that the horticulture theory and practices class. We do our labs in there, our lectures in there pre-COVID. And then we also have the plant propagation class. I also teach the seed science class, so I also use the facility for that, for demonstration purposes. And then Dr. Amore's class, TPSS 402, which is the flower and foliage production class, they also hold classes over there, and also the hands-on labs. Aside from that, the weed science class also comes in there. Several other classes from TPSS come in there. But I guess for us, we're the regulars, the 364, 420, the foliage class. So we're kind of regular there. And then the rest of the TPSS classes they come in there once in a while.
Can I come take classes from you? This
Sure.
sounds something really interesting, being a plant nerd like I am. The level of science and integration with the plants, it just seems amazing. And also, as a board member of HFNA, I was wondering, how can we help support your projects at Magoon? Dr. Amore?
I would say that HFNA, keep on doing what you do, because HFNA has been very supportive. We are extremely thankful for the support HFNA has provided for us. And I'd also like to add that HFNA has been very supportive in writing letters of support, which are usually included in the application packets for grants. And for the ongoing support and continuous support we've received over the years, we are thankful to have HFNA as an extremely supportive partner.
Well, we're glad to help. Well, we appreciate your time and all the effort this took. I'm sure you're extremely busy. So from all of us at HFNA, we'd really like to thank you. If you'd like to say maybe a little closing before we're going to break for lunch here in a little bit. So Dr. Baldos, while we still have you, if you want to just maybe say a few closing words for us.
I don't know. Well, you can, I guess, drop us an email, or if you want to, maybe if you're in town and you want to take a look at the facility, you can just email us and maybe we could tour you.
Wonderful. Dr. Amore?
I'd like to say big mahalo. Thank you to HFNA. Truly, you all have been partners in our research journey, and this continued conversation with growers has enabled us to make more meaningful research, knowing that we're helping each other. So it's not just one way that we're doing research, but truly the feedback from the growers, and like this opportunity to have feedback also. I hope the designers will also make comments as they see the various designs as to which ones are interesting to them. That will help open up research possibilities for us. So it is truly I really feel thankful that this partnership is available and we have this. Not many researchers, I think, have this ongoing, constant communication between researchers and producers. So I feel this is something that we should be continuing, and we're doing it. I think we're doing it well, and it's something that we should keep up with. Thank you.
Once again, we really appreciate your time and your expertise and the tour of your beautiful facility. And as I said, I'm jealous and want to come play with you sometime with all your plants. So have a great Aloha Friday.